Has what next to it?
TBA.
TBA. No, I would say to be. TBA. I'm not sure what that is. That like showing for the how it's going to be taught next semester? Are you looking at next semester's classes?
No,
Or
it says how it's going to be taught. It just says
okay.
It doesn't have a time or a date.
Oh, maybe it's like to be determined or to be announced to, yeah, something like that. Which class is it?
Microbiology lab.
Okay, yeah, they might still be working on fiddling with that. So that reminds me of some announcements I need to make for this class as well too. Because normally we have Biochem 2 in the spring, but at the moment we're not scheduled to teach Biochem 2, which most people, probably like 2/3, 3/4 of the class, like, oh, Biochem 1 is enough. But I do get some people who take Biochem 2, and we're not going to have it. Well, it's still kind of up in the air, but at the moment we're not. But I think if there were enough people who wanted to take it, it probably would be.
Don't you need it for the ACS certified?
Yes. But I don't think we have anybody on that ACS biochem track at the moment. So, because we were talking about this yesterday in a faculty meeting, it's the hardest track. It's the most intensive. It requires the most courses. So a lot of people don't want to take that course because it's just a lot of stuff. So we're working on finding ways to enhance our course offerings, to streamline the curriculum and all that good stuff there as well too, and figure out ways to make it so it's not so arduous, because biochem is definitely the coolest part of chemistry. Definitely. At least that's been my life experience. Okay. Other stuff that I should get to ahead of time here, and I'll put this announcement. So lab tomorrow. I will be out of town tomorrow, so we'll do lab remotely. Basically, I think it's the microscopy one. I'll just open it up and you guys will be able to go in there and do that. I might have if you're lucky, I'll have a pre-recorded like 15-minute introduction to it, but you might just be like, hey, see how you can do on this lab. So, but it shouldn't be too bad. Again, it's through Labster, so that, for most of Labster, all the stuff I've kind of played around with, most of it I've really liked and enjoyed. I know the carbohydrate one was probably a little easy last week's.
Yeah, that's the
RNA. Okay, yeah. So this one must be the RNA one that we're this week. Yeah. Okay. I was getting ahead of myself. I was thinking of next week's, which is microscopy, which I need to look at in more detail for next week. But anyway, yeah. So that's that'll be a good thing there too. So I will probably open up all the labs, if not by this weekend, by Monday, so that if you got nothing else to do, you can just do all the labs and be done. Yeah. All right. I always have to tell my story about self-paced psychology. Well, yeah, okay, good. We are recording. I set it to automatically record. When I was an undergrad, we had self-paced psychology, which basically you had to go in and take a test. I think you had to cover like 25 units in a semester, and you'd go take a test after each unit. And people would like this class because the idea was you could get it done and then have the rest of your semester to go do fun stuff. Not that I went to a party school or anything. But so many people who would do this would put it off and put it off and put it off, and then they'd be struggling. And like, how do you fail self-paced psychology? Because it's pretty much an easy A if you just went and did everything ahead of time. But people would get way behind and try and do everything the last 2 weeks, and that makes it really, really hard. So people knowing me were like, you're going to be in that latter group where you don't get it done. Nope, I was done in October. I was like, all right, time to go do fun stuff. So I don't know if that was a semester that I had 23 credit hours or not, but it was like, let's get all this stuff done. Yeah, all right, okay. Anyway, speaking of getting stuff done, ooh, like that transition? Oh, okay. I might be on today, might not. We're starting a new chapter here. I know the exam is there. I will get to it at some point. That is not the thing that I want. Where's the thing that I want? I must have minimized it. There it is. Okay. Yeah. Let's go back here. There we go. All right. So we will be transitioning into our exam 4 material, starting off with chapters 8 and 9. So this is all about nucleic acids. I kind of look at this as the last of the structures, because after this, we start talking about the cell as a whole entity and signaling and some fun stuff like that. But we have some structures and interactions here to deal with. So I know I break up chapters 7 and 10 because lipids and carbohydrates, sort of semi-fit together. But we're going to kind of, in some ways, as we get through nucleic acids, especially in this chapter, review a little bit of those carbohydrates, okay? So, because our nucleic acids are basically ribose derivatives. Okay, so I'll pull this up here. I swear I'm going to try not to be too lazy sitting in here. Although, this is a very warm room. It's only 85 degrees here right now. It was like 90 something in here yesterday afternoon for my lecture. I was dying. Yeah. So, yeah, choose your poison, I guess. Okay. So our nucleic acids and our nucleotides are what we're looking at here. We have, again, our regular goals in here, figure out their function, know their structures, understand the structure of double-stranded DNA, and talk about the structure of RNA a little bit. We'll talk about denaturization and annealing of DNA. And we'll talk about mutagenesis at the very end, just very briefly, just kind of introduction in there, a little bit of the chemistry of our nucleic acids. Okay, so our nucleotides and our nucleic acids, and we'll kind of get into a definition there as well too, are used for a variety of purposes within an organism. Mainly when we think of DNA, we think of the storage of genetic info, and that's certainly a huge major role. To transmit it, we need our mRNA. There's a great little video. I think I've got it posted for you guys in this class. I know it's definitely for second semester biochem. If we have it, we'll tier for me. I like teaching it. It's fun. But we can also have some nucleic acids that function as catalysts as well too. These are our ribozymes. So we have, a non-protein enzyme. Of course, we have our tRNA and rRNAs for protein synthesis. But besides all this cell information, and expression, we can also have monomers that are used for energy. ATP, GTP, CTP, and even TTP and UTP for lesser extent can provide lots of energy when you hydrolyze the triphosphate bond. We'll see a lot of enzyme cofactors. Our NAD, NADH, NADP, and NADPH are important cofactors. And of course, we'll see a lot of signal transduction, our cyclic AMP, right? And we'll see why it's cyclic AMP, I think, at the end of this chapter. All right, but first, let's start with some basic definitions. All right, so our nucleotide, we should probably start from the bottom. Our we have basically our I guess our nucleobase, all right, which is just the purine or pyrimidine ring by itself. Our nucleoside is going to be that base plus the pentose as well too, okay? And then our nucleotide has a phosphate group attached to it. So the way I remember this is nucleoside is second, so it's got two things, and nucleotide, T for 3, it's got 3 things in there, okay? And our nucleic acid is just, the ring by itself there, okay? All right, so we have these ring structures, all right? So our pyrimidine or purine ring, we number them in a certain way. We number our ribose ring as well too, but the ribose ring, we give those carbons, like 1', 2', 3'. So this is why you may have ordered stuff from like a 5' to 3', or if you've done anything in biology lab, 5' to 3'. That's where those primes come from. They're indicating that those are the sugar carbons, whereas like carbon 1, or in this case nitrogen 1, is the nucleic acid itself. All right. Let's see. Yeah. So yeah, and that just helps to avoid confusion and tell you which part of the structure you're dealing with. Okay. So the phosphate group is negatively charged at neutral pH. So that means, if you take DNA and put it in a cup of water, you're going to have a bunch of negatively charged stuff in there. The phosphate group is typically attached to the 5' position, but it can attach to any of those alcohols in there. And we'll see some neat variations on that as well. The nucleic acids are built using these 5' triphosphates. So our ATP, GTP, TTP, and CTP, we need lots of those in there so that we can attach those A, G, T, or Cs as needed. When we talk about PCR in lab, maybe that's the one that's 2 weeks from now. I can't remember my order at the end of the semester. It's been a long semester, it seems like. But you always have to have lots of nucleotides in there so that you have the raw materials to hook them together. If I go back to my Lego example, these are the little small Lego pieces that you attach together there. Okay. But again, when you attach them to the phosphates leave, so you get pyrophosphate as a leaving group, and that phosphate, you make a phosphodiester bond as you attach these. And we'll see more about that when we start talking about the polymerization. All right. We can have that phosphate in different positions. So typically, the way we're going to draw it is going to be here in the upper left-hand corner with our 5' monophosphate. In this case, we have our adenine, adenosine 5' monophosphate. But we could hook that here at the 3' hydroxyl group to have adenosine 3' monophosphate, or our 2' monophosphate. Or we could have this 2', 3' cyclic monophosphate. So you've noticed we've made another ring structure in here. This is actually, we won't get into this, but this is actually kind of a fascinating little intermediate in a particular type of reaction that occurs in the body. But it's just kind of neat how you can play around and make these different structures. It's possible to do this. You'll also note that comparing, here we have our sorry, this is not our deoxy, this is our ribonucleic acids. If we have deoxy, it's the 2' that lacks the hydroxyl group, so we can't form this structure in DNA, but we can in RNA. So part of our RNA degradation might involve that intermediate. All right. So the pentose forms can differ around there. We're typically going to have this furan ring. We're going to have a 5-membered ring structure in here with an oxygen in there. You can see we have one of our methyl groups kind of hanging off the top there. We can also, since we have a 5-membered ring and we don't have any conjugated bond systems in there, it's mostly planar, but it's not completely planar. We can have exo and endo puckers to them. So we can have a C2 endo and exo where the carbon 2 is kind of either popped up or popped down. I always think of that endo as being, oh, I got to go in endo, and you got to go inside it as it's puckered up that way. And exo is kind of puckered away from you. And we can also have our C3 endo and exo as well too. All right, looking at our nitrogenous bases, our nucleobases in here, we have 2 main forms, pyrimidines and purines in there. These are basically nitrogen-containing rings. These do tend to be planar. Right, you can see we have a lot of conjugated double bonds in here. So we can see that there's probably a good resonance structure in here as well too, kind of like a benzene ring. So this, depending on our substituents, this won't necessarily always be true, but these do tend to be very planar. And our nucleobases tend to absorb UV light somewhere between 250 and 270, which means that it's very handy for us to check for the presence of DNA. And we can quantify it based on the amount of absorbance at 260. Okay. Our pyrimidine bases, we have 5 that we're going to be concerned with. So we have cytosine, adenine, and guanine, both the DNA and RNA. We have thymine, only in DNA, and uracil, only in RNA. Again, stuff that you probably know. All these are excellent at hydrogen bonding, so they make good hydrogen bonding partners. So of course, that's how our double-stranded DNA gets held together by hydrogen bonds between the bases. There are base pairing. Okay? This is where the term base pairing comes from. Okay? So adenine are our purines. And then for me, I could never remember what's a purine, what's a pyrimidine. Well, it's the counterintuitive part. Purine, shorter word, bigger molecule. Pyrimidine, bigger word, shorter molecule. So it's like the opposite of what you expect, like everything inside. No. All right. But basically, these are structures that you should know. Maybe I'll give you something like this and say, hey, what's this molecule? But you can see our adenine is a purine with an amino group coming off the carbon up there. I think it's carbon 9, maybe. Yeah. And our guanine has a carbonyl group up there, and it also has an amino group coming off this carbon too. If we look at cytosine, thymine, and uracil, these are our pyrimidines. So we just have the 6-membered ring in here. Okay, and you can see cytosine has an amino group at the top and a carbonyl off to the side there. So it's kind of the opposite of the ring structure of guanine here, if you don't count that part over there. So G and C are kind of opposites of each other. That makes sense when you think that G pairs with C a little bit. So that might be a mnemonic that can help you. Our thymine, we basically have 2 oxygens coming off of here plus a methyl group. So I think it's a little weird in there. And uracil is basically thymine that has been demethylated. All right. So we can think here's our nucleoside. Okay, so adenine, guanine, cytosine, thymine, and uracil, or uridine, I guess, would be our nucleosides because they're just the bases by themselves. Sorry, no, sorry. Step back, nucleoside attached to the phosphates. This would be the nucleobase is adenine, guanine, cytosine, thymine, uracil. When we attach them to the sugar, we get the adenosine, and we add the sign to it. It's a sign, the sugar S sign, tooth in a second. I don't know, whatever mnemonic works for you. And a nucleotide, since we're adding that phosphate group to it, we put an 8 suffix on there to indicate that. So uridylate or adenylate. So that's why we have adenylation. If we add an adenylate group to say a protein or a lipid or something like that, adenylation is a modification that we can have. So we can put that up there. And of course, if we have DNA, we have the deoxy form. That 2' carbon is lacking the OH group. It just has a hydrogen there. So that would be our deoxy. So DNA, all sorts of stuff like that. Okay. So I like this. It's like you need to know structures, names, and symbols, both the 2-letter code and the 4-letter code. So, deoxy, adenine, deoxy, adenosine monophosphate. So the stuff that we have there, all sorts of good stuff there, right? All right, so these are our basic structures. And you can see, again, the good thing is for our ring structure, our phosphate group, it's all the same. Phosphate group's all the same. Ribose, all the same here. And then up here, the only thing we change are our bases, okay? So adenosine and guanosine are our purines, and these are our pyrimidines. And this, of course, is our DNA. So, right, if we take a look at RNA, very similar for adenosine and guanosine. Just we have that hydroxyl group there, uridine and solidolate as well too. Okay, so there's 8 structures, but really it's more like 5. You just have to remember whether you have this hydroxyl group here or not. Okay. And you can see this is monophosphate. I can stick another one on here and have diphosphate, ADP or GDP, or 3 on there and have triphosphate. Okay. So those are our structures. Should have brought my Mountain Dew with me. Yeah. This time of year, and I get the whole sinus thing going on too, which it's always fun because I have I'm part of a study, monitoring people for COVID and whatnot. So I have to answer my symptoms every day. It's like, well, yeah, I have congestion, but it's fall. So no, I don't have any different congestion. It's the only symptom I have. So, all right. Anyway, back to our DNA and RNA. In our nucleotides, this pentose ring gets attached to the nitrogenous base through the nitrogen group, so that we make an N-glycosidic bond. Okay, so the sugar is attached to a nitrogen in there. This bond gets attached to the anomeric carbon of the sugar in its beta conformation. Okay, so this is position N1 in pyrimidines and N9 in the purines. Okay, the bond's very stable towards hydrolysis, but we can cleave it in acidic conditions. Okay, so here's what we see. So if we take our nucleosides here, all right, here's our attachment to our anomeric carbon. That's kind of why we draw them all this way very consistently. And then our nucleobases are free to rotate around that bond. So that beta linkage, they can just kind of rotate around there. So if this is, say, like cytidine, here's my oxygen for my thumb, I can have it over this way. can rotate around that way as well too. Okay, so we've got full rotation. If, like in the middle here, where our bond angle, these are basically on opposite sides of the beta bond from each other, that's going to be our anti-conformation, which is going to end up being the most stable because we'll have less steric interference. Okay, you can see that we have this a CH2OH group over here, especially if you have something coming off here, like say an oxygen, you can get some steric interference. So the anti-conformation is the most stable, but you can have the syn conformation in there as well too, depending on what all is going on. Okay. All right. And then this is just kind of a ball and stick model in there to kind of show how you can kind of rotate this stuff around. All right. Got lots of bond angles in there. Okay, we got some other funky stuff that can happen here. Our nitrogenous bases can undergo a lot of tautomerism. So we draw in that certain way, and that might be one way to memorize them, but they're pretty fluid and dynamic. We can see a lot of keto-enol tautomerism. We can pass along a lot of protons in there. So basically, these are examples of prototropic tautomers. So keto-enol tautomerism, and then lactam-lactim tautomerism. Ooh, don't speak too fast. Both of these will exist in solution, but we'll see the lactam forms that are most common at neutral pH. All right. So here's our lactam. All right, where we have our carbonyl groups here. If we shift our proton from here up to the oxygen and drop this double bond down here between the carbon and nitrogen, we get our lactim. And if we do this with the other pair down here, we get a double lactam. It's like a double rainbow. That meme is what, 10 years old now. You guys curious how they got the name for this? So there's a research group out there. They were Scottish, but they had one American. His name was Tim, and he had to leave. So when they discovered this, they called it the lactim. And this is a lactam because it was wearing a Scottish. I'm totally lying. I just wanted to make a pun about Tams and Tims. Lactim. Tim's not here. We're talking about anyway. I'm trying to have fun. But anyway, our lactam and our lactim, I guess we're looking at amino groups as opposed to amino groups. All right. If we look at these nucleobases, because of those ring structures, okay, ring structure and double bonds in here, they're great for absorbing UV light. So, we get our pi to pi star electronic transitions. Hey, remember organic chemistry? Typically, this is between 250 and 270. We've got a great graph showing why that is, and we often measure them at 260. All right. The neat thing about this is that when you do excite the electrons in there, they decay very rapidly without emitting radiation. So that is a useful trait to protect genetic material from UV light. Okay. And there's no fluorescence in there. If we look at our different nucleobases, where they absorb at, you can see if we were interested primarily in GMP, we'd probably measure at 250. Okay. If we were interested in primarily in CMP or TMP, we would measure at 270. But we measure at 260 because that covers everything. Over here at 270, we're on kind of a downslope for our GMP and our AMP, so we won't measure as much. Same thing with 250, we're on a downslope for CMP, TMP, and even pretty much for UMP. So we measure at 260. That gives us the highest average absorbance for all 5 different nucleobases. Okay. All right. Yeah. Okay. So besides those 5, there are others that exist out there. Some of them are modifications that are done after the formation, and others are variations in there. Methylcytosine is very common in eukaryotes, also found in bacteria. So we do have this methylation pattern in here. Our N6-methyladenosine is common in bacteria, but not in eukaryotes. Why do we see a lot of these modifications that are made? A lot of them can be epigenetic markers. So, you can mark the DNA so that cells can degrade foreign attackers, like our prokaryotes. We can say, hey, we want to have this pattern. If we don't have this pattern, it doesn't belong here. Okay. It always makes me think when I was younger and going out to Pittsburgh and I'd see like parking lots that said no foreign cars allowed. The other thing we can do when we methylate something, it can be a way to mark what genes should be active. So we can kind of turn on genes or turn off genes depending on whether they have been modified or not. Can we turn them on and turn them off in an inheritable manner? Okay, this is a great question, which I'll go ahead and answer. It's like, yeah, sort of, kind of, somewhat. So, it was always fascinating. I always hate to talk about this in an election year, but if you think of eugenics and the idea of creating, the best possible human, eugenics often got transferred into, different sorts of racism, like the whole idea of the Aryan superiority was eugenics, and you'd have programs where you want to have like the best people in there. So eugenics has a very, very bad name, especially dealing with humans. Epigenetics, though, is not quite the same, but it's related enough to where when politicians start talking about, oh, we can't do this because, that's what their party stands for. We must do this. It's like, oh, someone who doesn't believe in eugenics but does believe in epigenetics. And there's a role for both. But our epigenetics are more of an environmental marker for kind of like what's going on. You can see this with, I think there's a plausible example is, like the elephant memory. Elephants who may have been born far away, but they know where they need to go for an oasis or something. It's kind of genetic memory, and it has to do with the modification patterns that somehow do get transferred. You could also say, if you look at like, it's like your daddy was a horse thief, and your granddaddy was a horse thief, and your great granddaddy was a horse thief, so you're probably a horse thief. I watched too many westerns as a kid. There's an epigenetic connection in there as well too. Yeah.
So is that why, like I saw this somewhere, is that why animals know what to do without being told compared to humans? Like you gotta really train people. The animals, they're not gonna walk by themselves, eat by themselves.
Possibly, maybe. we're pushing the limits of what I know that you. But there is an epigenetic component to some animal traits. Yeah, but some of that could be. I'm sure a lot of that is other stuff as well too. So there's a lot of stuff that kind of combines together there to get that what's the term? Giving my brain here too. Our whatever the term is for the natural inclination to do something that way. But this would be more like this particular pattern of genes is going to be being expressed down through different offspring. I was teasing my wife last night because my daughter desperately wants new shoes. She's like, my shoes are too small. I need bigger shoes. And it turns out they actually were. And she can wear the bigger shoes that we had. So she's all excited about that. I need shoes. I need shoes. And my wife is complaining, why? Because she wants lots of shoes. And I was like, you cannot say anything, because I pointed over to the closet that had like 2 shoe storage things full of shoes that she probably hasn't worn in years or something. And I was like, yeah, the apple doesn't fall far from the tree, does it? Mm-hmm. Yeah, so there we go. All right. Anyway, we also have a variety of others. So you can see here we have some different methyl compounds in here. You can even see we can make this more complex. In the bottom right, we have a hydroxymethylcytosine. So we can do a hydroxymethylation. We can also see some other minor nucleosides. If we look at RNA, we get a lot more variation. I always laugh at this description from this slide. Inosine is often found in a wobble position. More on that later, much later, in the anticodon and tRNA. So it's going to be in that 3rd position in there. If you deaminate adenosine, you get inosine. Okay. And the explanation, it provides a richer genetic code. It's like, yeah, I add butter to the gravy to make a richer taste experience. But I think what they're trying to say here is that, it allows for some variation. That wobble position, it's like, oh, you can fit something in there that's not quite supposed to fit there. All right. Pseudouridine is a very common RNA minor nucleoside as well too. Basically, we isomerize uridine in there, and it probably helps stabilize some structures in there. So it's conformationally important, and you'll see some conserved pseudouridines in certain positions in mRNAs and tRNAs. So that's what they look like. Again, you don't need to memorize these. You got some crazy things like thiouridine. Instead of an oxygen, we put a sulfur in there. You know, methylguanosine plays a pretty important role. But anyway, those are all our building pieces. All right. Oops, can't lose that. Now we have it on record of my wife interrupting yet again. Yes. How many times? Yeah. So like, I don't have many hours where I'm teaching, where I'm actually physically in class, but yes. Anyway, need to put like a big, never mind. Need to post it over the shoes so she'll read now. Anyway, she finds out how much to talk about her in lecture. We go, oh, all right. Our polynucleotides, we can start hooking these together and make interesting structures. Okay. So again, we're going to have these phosphodiester linkages that phosphate in the nucleotide plays that critical role because it's going to be the linkage between, say, our A and our G and our T and our C and so on and so forth. DNA backbone is really stable. You can find DNA from mammoths. I believe that they've tried this experiment where they take an egg from like bison and take out the DNA from the bison and insert mammoth DNA. See if you can get mammoths. Jurassic Park, right? Jurassic World. Woo! All right, we can make dinosaurs. All right. But we do have some various enzymes that will cleave these backbones. Our DNases. But again, this is why they say DNA is forever. So if you like licked a postage stamp when you were 12 and then commit a crime when you're 50 and they find the stamp, you can link you. That's where the whole forensic science and CSI effect comes in because we can store DNA forever. RNA, on the other hand, really unstable. You look at it, funny, and it will degrade. It can last for a few years in water, but in our cells, it's typically degraded within a few hours. All right. If you store it properly, I guess it'll last a while. I was going to say a few years. I'm like, yeah, I don't know about that. Our polynucleotides are all linear, so no branching. So again, only carbohydrates seem to branch. Our directionality, our 5' end is different from our 3' end. So we always read from 5' to 3'. If we look at RNA, unstable under alkaline conditions, there are also RNases everywhere that will chew up RNA. So sRNA in plants will prevent various inbreeding. RNase P is a ribozyme that processes the tRNA precursors. We have dicer. You may have heard about this because we can use this for RNA interference. So basically we have these interfering RNAs that will prevent gene expression that can be used to treat genetic diseases. Dicer can also be protective from viral genomes as well too. All right. So how does this work? Yay, chemistry. Okay, so what we've got up here, we have an RNA base linkage in here. So this hydroxyl group here is actually, we're going to see a base catalyzation. Our base is going to abstract off this proton, and then that lone pair is going to come down here and attack the phosphate. And when it does that, we form this 2,3 cyclic monophosphate group as we cleave off the rest of the base, the rest of the RNA sequence in there. So we get this intermediate that we kind of saw in the beginning in there. So the presence of this hydroxyl group makes this inherently less stable, makes DNA easier, RNA easier to get chewed up. DNA does not have that instability because without that 2' hydroxyl group, you can't undergo this reaction. So DNA sticks around a lot longer. All right. Were we all in person for our exams? I'd certainly say draw a phosphodiester linkage from 5' and 3' between, one nucleotide here and another nucleotide here. Pick two nucleotides. I don't know. Yeah, show me this linkage. But this is how it works. Here's our 5'. Here's our 3'. So we go from our 5' and 3'. I'd read this as ATG, and here's our linkage. Okay. Same way with our RNA. I'd read this as UGC. Okay. So I know those of you in chemistry who will have senior seminar will, if you haven't already, you'll get the question. There will be a question at the end that says, where the answer will be hydrogen bonding. So our hydrogen bonding interactions are very important. Even though these are very weak interactions, our different nucleobases can hydrogen bond with each other to make that base pair. So A pairs with T and C pairs with G. And the idea here is we have purine pairs with pyrimidines. Okay. All righty. So if we look at something like this, our AT, you can see here we have our purine and our pyrimidine that are base pairing, and we make 2 hydrogen bonds. Okay? If you look at our GC, again, you can see that our cytosine is kind of the opposite. Our oxygen amino group, carbonyl amino group ends up in there. So we actually end up making 3 hydrogen bonds between G and C. So which one is going to be harder to break apart? Yeah, GC will be harder to break apart because you got 3 hydrogen bonds instead of 2. So that becomes important when we talk about denaturization of our DNA, and when we get the replication forks and stuff like that. So melting points and all that good stuff in there. All right, but this is our general base pairing. Good thing to know, AT, we got our 2 hydrogen bond pairs. GC, we've got 3, okay? So let's take a moment and talk about the discovery of the DNA structure because it shows up in social media like every year. So it's, again, one of our most important, this was a huge step forward for biochem because previously it was like, hey, how do we make beer? What about sheep's wool? And now all of a sudden it's like, oh, DNA. So we get back to the 50s and Watson and Crick, very important, and Franklin and Wilkins also critically important for this. All right, so there are lots of missteps in here, lots of different knowledge, lots of collaboration. Some would say data stealing, the cost of sharing your data too early. So we got Miescher, who comes up with this, nuclein from the cell nuclei, chemical analysis. See, we got some stuff going on here. So Franklin and Wilkins were the ones who worked on the X-ray crystallography of DNA, which I love this. That's the X-ray crystallography pattern that you got to work with if you were early into science. Now you have like thousands of images in there, but yeah. But the cross basically means, hey, we got some type of helix. The diamonds indicate we've got maybe a phosphate sugar backbone on the outside. And they figure out the parameters of the helix. Watson and Crick were looking at DNA. They figured out the hydrogen bonding, and in this missing layer, we had this alternating pattern of major and minor grooves in here so that the double helix would fit the data that paired with them, and they got the Nobel Prize for that. Now, you often see women in science will say, Rosalind Franklin was screwed out of her Nobel Prize because she was a woman. Well, unfortunately, she was screwed out of her Nobel Prize because she died. Had she not died in a car accident at the age of 38 or 39, she would have been awarded the Nobel Prize as well, too, but they don't do it posthumously. But still, she's an example of an early woman in science. She had other issues that she dealt with as a woman in science, which are more valuable than the Nobel Prize thing there, too. So, and then, of course, if you win a Nobel Prize, you can go on in later life to make incredibly strange pronouncements. Like, I believe it was Watson who was like, I feel sorry when obese people come for a job interview because I know I'm not going to hire them. And I think he's got some, yeah. So he kind of gets out there as well too. So the point is, what's that?
He's alive.
Yes, I believe so. I believe he's still alive. Yeah, he may have. Well, these slides are like 2 or 3 years old. I can't remember, but yeah. Oh yeah. Well, Watson was a little bit younger than Crick, but yeah. But I think Watson is like 98 or he's quite old. Yeah, you can do a quick Google search in there too. Yeah. And of course, you got these guys here as well too. It's like I always feel bad for Wilkins because nobody ever talks about him in no case. But he just looks sad there too. It's like nobody's going to talk about me. He's like the Eeyore of the group. Anyway, yeah. All right. So it turns out though that we actually have multiple forms of DNA. So we do have this helix, but we can have multiple ways we can actually make this helix. And the way it ends up, primarily is in this form we call bDNA. You have a major groove that's a little bit further apart and a minor groove that's closer together as these wrap around each other. You might recall that we talked about a particular protein, the alpha helix, is just the right size to fit in the minor groove. Yeah, so we can see our protein DNA interactions revolve around minor groove interacting with alpha helix. There's a compressed form of DNA known as the A form, where it's a little bit shorter. And there is a kind of inverted, unwound, stretched out form of DNA known as zDNA. I believe zDNA primarily occurs when you dehydrate it. So if you take DNA out of a liquid solution and dry out all the water, you end up with this kind of weird stretched out version of it. But bDNA is the important one for us. We'll kind of keep that one in there. Not too compressed, not too worn out. Our B form is a right-handed helix. There's about 10.5 base pairs per turn. The rise to run is 3.4, so that might sound familiar to like our alpha helix with proteins. The other thing is we have this anti-conformation for our glycosal bonds in here. This differs with our Z form where you have syn for the purines. So the purines are folded back in on themselves as you stretch them out. All right. So in order to make our helix, we have two complementary DNA strands. So we have two chains that differ in their sequence. Keep in mind we read from 5' to 3'. So this first one we can just read down, but the second one we read from the bottom up. Okay, but you can see the complementarity. Our ATs and our CGs in here are going to pass that way. So what this means is we can say these two chains run anti-parallel. One, we start at one end to read one, we start at the other end to read the other one. Okay. All right. So how do we replicate these? All right. Well, we unwind our helix. Okay. So we got this nice little strand separation here. In blue would be our parent strand, our mother strand, and the ones in red will be our daughter strands, our newer ones. So each strand is going to serve as a template for the synthesis of a new strand. We have DNA polymerases that catalyze this. You get lots more of this in Biochem too. So basically we have a new DNA molecule. One of the strands is from the parent and one is from the daughter. This is a little easier than, say, taking a parent strand, unwinding it, copying it, making two new daughter strands, and then putting the parent strand back together and the daughter strand back together. This way, with this semi-conservative replication, where we hold on to one parent strand and have one new daughter strand, if an error is made in replication, then a proofreading enzyme can come through and kind of look at it, compare the two strands and say, hey, these bases don't match up here. It will check and find the parent strand and it will cut and fix the daughter strand to match the parent strand. How does it know the parent strand? Methylation. So that post-replication modification helps indicate which is the parent strand. So we're going to assume the parent is correct and the child is wrong. Like, I need new shoes. So the methylation process happens after replication. So another enzyme will come through and find the daughter strand and methylate it in its proper position. But until then, the proofreading comes in first and kind of looks at that and makes sure everything matches up. Okay. That's a lot to try and cram into one slide. I always want to try and lecture too much with too much because biochem too. I don't know. Yeah. Anyway, anywho, we can talk about messenger RNA as well too. Our mRNA is probably the one that gets talked about the most. It's the code carrier for the sequence of proteins. So if we go back to our universal dogma from like chapter 1, the DNA is our repository of information. The mRNA is the expression of that, and then our proteins are our physical expression. I guess the mRNA is our translation. Okay. All right. So mRNA is synthesized using DNA as a template, and then generally mRNA is single-stranded. You can have double-stranded mRNA. It's just a little rarer. In fact, one of the things when I was bored, I took like 3 books to Indonesia for like a month-and-a-half long trip one time. And one of them was the Lippincott's Illustrated Review to Microbiology. So I read about different viruses that have single-stranded and double-stranded DNA and RNA, and some that are encapsulated and some that are not. So it's fascinating, but you can have double-stranded RNA. But most of the time we're going to talk about single-stranded. Again, we have ribose instead of that deoxyribose and uracil instead of thymine. The neat thing is, our DNA is only going to code for one mRNA, but our mRNA might code for more than one protein. So we can have, polycistronic, and this is for mainly bacteria. In eukaryotes, we see alternative splicing. through our mRNAs. So we only get one gene product, but it depends on how we cut up our mRNA as well too, to assemble it into the, to translate it into the protein. So mRNA and tRNA transfer genetic information from the DNA to make our proteins. Okay. All right, so there's again our nice little single strand mRNA. Okay, so in terms of our DNA, monocistronic, one strand of DNA codes for one gene. Okay, in there, and that's typically what we have in eukaryotes. Right, bacteria, because they're much smaller, our prokaryotes will often be polycistronic because they have to cram more information in there. And a lot of this can deal with the reading frame, or we may have some series of linked genes in here as well too, but they'll have multiple genes per nucleic acid. Okay. So yeah, we tend to be monocistronic, eukaryotes tend to be polycistronic. Okay. When we look at our single-stranded RNA, we can have some issues that arise. We can have palindromes in the sequence. So, palindromes are just words that are the same forwards and backwards, like civic or race car or rotator. I always think of radar. I'm sure you guys all read Finnish and Greek, so, there you go. I am the Finnish word for soap cup batch trader. Really? Is that a thing in Finland? I need to find some Finnish graduate students, international students, and ask them how many soap cup batch traders they know. That's crazy. Anyway, but when we get to our strands of DNA or RNA, our nucleic acids, if we have a palindrome, those mirror repeats can kind of throw off the interaction. So instead of maybe matching up here, maybe they match up backwards, and that's bad. Okay, I'm gonna do my old George Bush impersonation from the 90s. We can also have mirror repeats. Okay, so we can have these things that are internally complementary. So we can have a single strand that binds to itself. If you ever design primers for PCR, which you'll probably talk about in one of the upcoming labs, then you want to avoid having palindromes and mirror repeats and long repeated, like poly T groups or poly A groups, because it's easy for that to hang up and make a mistake and not bind to your DNA properly, bind to itself or bind to something wrong. It can interrupt. And these are often used. So for example, here, if we have a nice little palindrome, they can perhaps bind to themselves. So instead of having a nice continuous thing here, we get a little region that's double-stranded. Okay, this is actually important for mRNAs because we will see a lot of double-stranded sections to make our cruciform structure in there. Okay, our RNA molecules end up having some really crazy-looking structures. Okay, so you can see here. Here's an example, and these little arms, will poke out, and they can often fold in on top of each other as well too. Okay, so these complex structures are stabilized by non-Watson-Crick base pair interactions. Okay, so we basically have other base pairs that are going to interact with each other and kind of stabilize things. We're not going to sweat that too much yet. Okay, so we just have, again, hydrogen bonding holds stuff together. It's like really weak glue, but if you put enough on there, it's going to stick together. At least hold your bumper on until you can get to the dealership. All right. Where are we? Just flying today. Okay. Alrighty. So, I'm gonna take a peek ahead here. No, I wish this would show me what I want to see here. No. Aha. Okay, it does show me what I want to see. All right. So I've got time to do a little bit more. So we'll talk about DNA denaturization as well too. So here we have our double strand that's wound around each other, and we can unwind it and separate that. We see that for replication, we see that for gene expression, all sorts of stuff there. Basically, if those DNA is hooked together, annealed together, you can't express genes. You have to unwind them to enable accessibility in there. So covalent bonds remain intact and the genetic code remains intact. When we denaturize it, we're not breaking the DNA or anything like that. We're just breaking the hydrogen bonds between the two different strands. We also lose this base stacking. So one of the ways that this helps get stabilized is our nucleobases. When they're kind of poking out this way, they can line up over top of each other. So you have this kind of vertical pi bond interaction that helps stabilize as well too. But when we denaturize the DNA, we lose that base stacking. So you'll see that UV absorbance increase because you now have more of these nucleobases available to absorb UV light. How can we denature them? High temperature, change in pH. Denaturization is usually reversible. So, if we heat them up, we can separate them into 2 strands, cool it down, and they'll go back together. Okay, so kind of like we saw with protein folding, was it cytochrome C? If you denature a protein, it unfolds, and if you dialyze away our uracil guanidinium hydroxychloride, the denaturing agent, it will fold back into its proper shape. So same thing happens with our DNA denaturization as well too. We'll often talk about thermal denaturization. We'll see this in PCR, which we'll talk about next week as well too in lecture, and then do that in lab. Our DNA is at a double helix at normal temperatures. We heat it up, we add enough energy that we're overcoming the energy of those hydrogen bonds. So just the heat energy. So in this room, my DNA is melting. Okay, not really. We can add enough heat, add enough energy in there to break those hydrogen bonds and dissociate them. And when we cool them down, they'll reanneal. So this is the basis of this polymerase chain reaction. All right, we'll talk more about that next week. We can monitor denaturization by the UV spectrophotometry, and we can see something neat here as well too. Different compositions of DNA will have different melting points. You can see the one in red, it's kind of all or nothing. You have basically this nice little strand here, you heat it up, it's good, 75, 80 degrees, we're good. We get to 85 and it just all unfolds. The one in blue is, maybe not quite as dramatic. It starts to unfold at 80, but by the time you get to 85, it unfolds. So we can look at where we have 50% denaturization. We'll call that our melting point. And for the one in red, it doesn't matter because, like 84, 86, you're, about the same spot here. But, for the one in blue, it don't matter. All right, so what affects our DNA denaturization melting point temperature? Okay, our CG percentage is in there. We mentioned with those 3 hydrogen bonds, look at that, 3 hydrogen bonds, there we go. That requires more energy to break apart. So if you have a higher percentage of CGs in your DNA, you're gonna have a higher melting point, okay? If you have longer DNA, you have more hydrogen bonds holding together, so you're gonna need more energy to kind of get it to denaturize, yeah. And of course, our pH and ionic strength. If we throw a bunch of salt in there, that's going to stabilize a lot of these reactions, so that'll increase our melting point. And if we change our pH, of course, we're changing our interactions as well. Okay. So we can kind of graph our melting point versus our GC content. And you can see that, if you have 75, 80% GC, you got to get above the boiling temperature of water to break that stuff apart, right? Conversely, if you're down to 1/3 GC, it'll melt at around 80. So you can see, no GC, you'll melt it at 68. Okay. It always troubles me that this graph doesn't go all the way up to, well, what if you have 100% GC, which doesn't exist in nature, but if you did, what would his melting point be? I guess we'd have to get the line equation here and extrapolate off. Yeah, put it in something that we won't boil. All right. So another important factor, denaturization is not uniform. We often talk about, and there are all sorts of jokes out there too, about unzipping the DNA, unzipping the genes, right? And you think of a zipper, you start at one end and zip to the other end, okay? But that's not what happens here. What we find is that AT-rich regions are going to denature it faster. So we'll have a little section there that'll take a little bubble. So that's how I know I'm talking too long. No, the Zoom's still working there. It's just that, okay, there we go. The room is like, no more lecture, that's enough. I'm sure everybody agrees. But we're almost done with what I wanted to cover today. But our red arrows here are pointing at our sites of denaturization. So these would be AT-rich regions where that strand is starting to separate. So we have a discontinuous denaturization. We start at the AT and pull them apart. So if you think of like zippers that, they came apart in the middle and you can kind of pull them apart that way. That's sort of what we have going on here. Our GC-rich are going to be the last ones to come apart. Okay. So we can utilize all this information in a variety of ways. Okay. We can detect specific DNA molecules. We can make a complementary strand and kind of probe to see if we find that sequence there somewhere. We can either have our probe be radioactive, which is really, really precise, but people don't like radioactivity, so we often use fluorescent DNA chips and just measure the light that comes from them. We can also amplify specific regions of DNA. If we make primers that surround the sequence we want to amplify, we can utilize the polymerase chain reaction to amplify that. We can also do something similar. We can make primers that almost match, but have like 1 or 2 base pairs that are wrong, and we can induce site-directed mutagenesis. So if we have primers that have an error in them, but they're still good enough to bind to the DNA, we can start amplifying that section of DNA and amplify a string of DNA with a mutation in it. Okay? We can also utilize DNA sequences to look at evolutionary relationships as well too, to see how things have developed or changed, whether within a species or across species in there as well too. And then of course the stuff that always interests me is like the antisense therapy. We can do like interfering RNAs to prevent a bad gene from being replicated, or to kind of turn off a gene that way. So this antisense therapy, I always like genetic modification as a medical treatment in there. So just because the room is telling me we should go and it's hot in here and it's been an hour, we'll end a little bit early today, but we'll finish up next week with mutagenesis and then start into chapter 9. Chapter 9 will tell us about various technologies and applications. So we'll talk about PCR in a little bit more detail. We'll talk about chip arrays, microarrays, various probings in there. So kind of like we saw, like ELISA for proteins, we have a whole battery of applications in here. We'll also talk about DNA, figuring out the sequencing DNA, the human genome, and we can talk about the Human Genome Project because human genomes, thousands of base pairs long. You really can't start at one end and go to the other, so they used a shotgun approach and then reconstituted it in there. We'll also talk about our cDNAs as well too, where we're just looking at the mRNA introns as well too. Sorry, exons. We'll talk about introns and exons a little bit as well. But anyway, that's for next week. Like I say, I won't be around tomorrow, so no lab meeting tomorrow. I might have a video for you guys, but we'll look at an RNA experiment, which seems kind of fun there. What else? I think that's all I want to say for this week, but have a good weekend, relax. If in lab you have outstanding things that are due to me, get them done and get them submitted. I've been getting the emails from the registrar. It's like, who is not capable of passing your class? Luckily, nobody in lecture lab for me in biochem is at that stage. I do have like a couple people who owe me multiple labs. I'm like, it's better get those done, But I have another class where I'm like, okay, I have like 2 or 3 students who there's just no way they can pass because they just haven't done anything. And it's like, yeah, I turned in the first assignment and then stopped. So yeah, yes. I probably won't get to, because I'll be out of town most of the weekend, I probably won't get to the test until Monday, but I should have it graded by Monday evening, most likely. So usually no. We'll see how things go. I had one year where this was also the year that I ended up going to Indonesia for 6 months, where I had a very anomalous chapter 5, exam 5, and I think it's because basically the students had an entire week without me before the exam and had someone else administer it, and I made it multiple choice. I had a guy in there who had a PhD from another country who was just taking biochem here as practice for getting the medal. I mean, he was on top of everything. He had like a 99 on every exam, and he scored in like the low 80s on that. and that was the highest grade on that. It's like, I need to look at this exam. So part of it may have just been, hey, we've got kind of like 2 weeks without Dr. Ebert. We're going to forget everything. Part of it may have just been it was a really hard exam. But I will occasionally make adjustments, but I try not to drop anything because I want to push everybody to learn everything. Yeah. But you may have some choices on upcoming exams, like answer 3 of these 4 questions or something. You got to figure out how to do that in Canvas.